Hi, my name is Aisha Arshad, and I'm a junior from Maine South. This past summer, I conducted an experiment testing the effects of different concentration levels of antimicrobial and antibacterial cleansers against Escherichia coli. I would like to begin my presentation by acknowledging the help that I have received throughout my research. I would like to thank Ms. Wagner, Maine South, and my family for supporting me throughout the process, as well as providing me with the opportunity to produce these results. When washing our hands, we aim to remove any and all harmful bacteria and germs. However, do all cleansers, specifically those claiming to be antimicrobial and antibacterial, work to achieve this goal successfully? If scientists can determine which antimicrobial cleansers work best to fight against harmful bacteria and germs, they can help reduce the number of infections inflicted upon the human population. Especially since the start of the pandemic, we have all been washing our hands more frequently in hopes of preventing infection. But will any cleanser eradicate the microbes that cause us to get sick? Scientists have done research to answer these questions. Almost all of the research publications extracted from scientific journals have had similar procedures of simulating hand washing with different cleansers, then introducing bacteria to them, and then incubating the solutions while monitoring bacterial growth over a set period of time. After a thorough analysis of these recent studies on cleansers against E. coli, the results concluded that cleansers with the highest concentration of antibacterial or antimicrobial chemicals perform better in the eradication of bacteria. Using this knowledge, I created my testable question, which was what are the effects of antimicrobial cleansers that contain triclosan or trichlocarbon and similar alternatives against Escherichia coli? Triclosan and trichlocarbon are chemicals that are the most commonly found in cleansers claiming to be antibacterial or antimicrobial. Continuing with this thought process, I hypothesized that cleansers containing the largest concentration of these chemicals would be the most effective against bacteria. My experiment focuses on determining which antimicrobial or antibacterial cleanser works best against Escherichia coli bacteria. By manipulating the independent variable, which is 3 different brands of liquid cleanser, 2 being antimicrobial and 1 being non-antimicrobial, and adding a fixed amount of Escherichia coli into each sample, I measured the effectiveness of each brand, which is also known as a dependent variable. The effectiveness of the cleansers was determined by examining the visible amount of Escherichia coli colonies. For the procedure of this experiment, cleansers with different amounts of antimicrobial and antibacterial chemicals were mixed with equal parts of water. They were then piped onto agar plates and streaked with a colony of Escherichia coli bacteria. These plates were then placed in an incubator for a total of 2 days and were photographed every 24 hours to monitor growth. This was also recreated with solely water as a control for the experiment, and 3 trials were conducted to ensure the accuracy of results. All of the materials used during my experiment are listed here. Some of the key materials include graduated cylinders, test tubes, cleansers, E. coli bacteria, and an incubator. This slide of photos shows the tested cleansers. These cleansers include an antimicrobial Clorox cleanser, an antibacterial Dial cleanser, and a non-antimicrobial or bacterial cleanser from Bath & Body Works. This is a photo of the solutions that were created to streak the plates. Each of them are equal parts water to cleanser. The independent variable in this experiment is the type of cleanser that was tested against the bacteria, and the dependent variable is the effectiveness of each cleanser, which was measured by counting the amount of E. coli bacteria colonies that were formed. These are pictures of the bacterial growth plates of the antimicrobial and antibacterial cleansers after 24 hours of incubation. Up here we have the Clorox plates, which, as you can see, are quite clear and have little visible colony growth. The cloudy patches seen on the plates are stains left over from the lather bubbles. Below those are the pictures of the Dial cleanser plates. Though there isn't much bacterial growth, there are still a couple of developed bacterial colonies in all 3 of the Dial petri dishes. These are pictures of the bacterial growth plates of the non-antimicrobial or bacterial cleanser and the water control after 24 hours of incubation. The cloudy streaks and patches seen on both sets photos are called lawns, which are many bacterial colonies grown together. Both of these variables had the most growth after the incubation period. This slide of photos shows the Clorox and Dial cleansers after the 48-hour incubation period. In both sets of plates, the bacterial growth had matured further and developed a couple of more colonies, but there wasn't an extreme amount of bacteria growing on either one of the tested cleansers. Finally, this slide of photos depicts the bacterial growth plates of the non-antimicrobial and antibacterial cleansers after 48 hours of incubation. These plates all followed the expected trend and matured further to form more bacteria. The colonies on both sets are more prominent and have grown more significantly because of the lack of chemicals to combat the growth of bacteria. Results and discussion. The graph right here displays the average number of bacterial colonies found in the 3 trials of the different cleansers. The significance of the different cleansers is essentially the difference of chemical concentrations in the solution. These chemicals are the reason for the effectiveness of the cleanser. As you can see, the Clorox and the Dial performed significantly better than the non-antimicrobial hand soap and the control of the water. This is because they contain chemical concentrations that eradicate bacteria effectively. When comparing the Clorox and Dial cleansers, there is also a slight difference in effectiveness. The Clorox performed better than the Dial because it contains a larger concentration of an antimicrobial chemical. As seen in this graph, the Clorox antimicrobial cleanser was quite effective in eradicating the Escherichia coli bacteria. These successful results were expected because the cleanser had the highest concentration of chlorothalonil at 0.75%, which is a chemical that has antimicrobial and antibacterial properties. The agar plates which were streaked with this cleanser had an average of 15 Escherichia coli colonies. The Dial antibacterial cleanser also received expected results. Because the cleanser has a lower concentration of benzalkonium chloride at 0.13% when compared to Clorox's cleanser, which had chlorothalonil at 0.75%, it was predicted to perform less successfully. This prediction was proven correct. Benzalkonium chloride is a chemical that is a common alternative to triclosan or triclocarbon, and it is also quite similar to chlorothalonil used in the Clorox cleanser. Despite this cleanser not being the most effective, it still displayed a decent job of removing the Escherichia coli bacteria. In reference to this graph, it is clear that the colonies of bacteria which were found on the plate still stayed relatively low, with an average of 47 colonies of bacteria per plate. Similarly, the non-antimicrobial cleanser had also performed as expected against the Escherichia coli bacteria. This cleanser was predicted to perform the worst out of all the tested cleansers, which this experiment proved. This was accurately predicted because of the lack of chemicals in the solution which have antimicrobial or antibacterial properties. Both the Clorox cleanser and the Dial cleanser contain these chemicals, which is why they performed far better than this cleanser. Referring to the graph on the right, it is displayed that the colonies of all 3 trials are in the mid to upper hundreds. The average number of colonies found from all 3 of the trials is 163. In comparison to the 15 colonies when testing the Clorox cleanser and the 47 colonies of bacteria found when testing the Dial cleanser, the amount of Escherichia coli bacteria found in this cleanser is remarkably high. A couple of errors that would confound the results of this experiment are differing amounts of cleanser water solution being piped onto the plates, error in streaking methods, and a lack of incorporation of E. coli with the cleanser water solution. The results from this experiment concluded that cleansers with higher amounts of antimicrobial or antibacterial chemicals are more efficient in the process of eradicating bacteria such as Escherichia coli, which was tested in this experiment. Essentially, these findings also revealed that which variables in cleansers contribute to the safety of humanity, as we rely on hand washing as a popular method to rid of germs. Thank you for your time.